Hello, everyone. Good morning for those of you joining in the US, and good evening for those of you from India. Let's start today's day. Let me share my screen. Welcome to the research day of our inaugural annual IndoUSrare conference. I am Reena Karta, one of the board directors at the organization. I oversee the research program. And today I will be co-hosting this session with our founder chairman, Dr. Harsha Rajasimha. So we kicked off our event yesterday with an advocacy day. We had an opening keynote address by Dr. Madhuliga Kabra, who is from the Indian Institute in the AIIMS in India. And then we had a lot of scintillating discussions by patient advocates. And today we will be focusing on research. And I would like to begin by thanking our sponsors, donors, volunteers, advocates, all our supporters who had been incredibly, very supportive of our program. And we are where we are today because of this immense support from this community. So I would like to first start with a quick overview about our organization. So it's very well known that there is a severe lack of global diversity, equity, and access to clinical trials. And at IndoUSrare, we work to increase diversity, bridge this gap. So we want to increase this diversity, equity, and inclusion globally, and accelerate development of therapies, not only patients in India or in US, but globally by building bridges to between these Eastern and Western silos. And we want to increase collaboration. And one way we are trying to do this is by this multitude of programs that we have launched. So you will be hearing more about the Patient Alliance program tomorrow and the Corporate Alliance on the following day, on the last day of our event. And today we also do have some awareness and education programs as well as policy advocacy programs. And today I'll be focusing on the research programs that we have been working on in the last 2 years. So we had collaborated with RareX and have contributed to publications like on patient-owned health data registries. And there's an upcoming perspective article on the impact of COVID-19 on rare diseases. As well as not only in the research area, but as well as the clinical development. So we wanted to give a global perspective. So we have been collaborating with institutes as well as individuals, both in India and US, to come up with these publications. And right now we are working on an incidence and prevalence study. And this is a study that has been supported by a health research grant from Takeda. And the idea is the aim is to scientifically estimate the prevalence and incidence of a subset of rare diseases in India, US, and globally. and to generate a scalable methodologies to expand this to other rare diseases. And this is the work we are again doing in collaboration. So collaboration is our key word. We try to collaborate with not only physicians or researchers, but all experts, including patient advocates in US as well as in India and also globally. And one major program that I wanted to talk about is our IndoUSrare Research Core, which is a multidisciplinary group of key opinion leaders, all stakeholders in the rare disease world. And it includes a disease-specific patient advocacy organization, both our member patient alliance members as well as non-members in India, US, and globally. And the goal is to accelerate the process of identifying patients with specific rare disease or a specific gene mutation, which maybe is very unique to India. And that way it can expand the community for a particular patient group who is looking for expanding their community. They could access patients in India, and also to better understand the geographic and ethnic differences in rare diseases, which is right now a major problem. That's where we have this lack of diversity. And so by identifying these local differences, by including patients from India, we try to expand our knowledge about a particular rare disease. Currently, we have about 50 members that includes physicians, researchers, and other experts from renowned centers and research institutes, both in India and the US. And we are open to any global collaboration. So if you are interested, please email us at admin@IndoUSrare.org. And one other example where we use this expertise as in our technology-enabled patient concierge program. So yesterday, Dr. Harsha Rajasim had talked about this, and idea is that we want to increase streamline the process of patient request intake. So when the patient approaches us, usually it is for either seeking a better advice, a second opinion, or maybe looking for any treatment information or any information about the disease. And we would like to pair them up with experts. And this is where we would like to utilize your expertise. And this is where we want to bring this research core program to. And I hope you will be open to such collaborations. And we are always looking for expanding our repertoire. So how can you engage with us? So again, we can, if we are a patient advocate, please apply for our membership. If you are an industry partner, apply for become our corporate alliance member, subscribe to our newsletters, discuss possible collaborations, partnering, and sponsoring. And there's always opportunity to volunteer and make referrals and introductions. And we are also active on social media. And again, you can find us on our website, as well as contact us at admin@IndoUSrare.org. So today, I just wanted to focus on research again, and we have an incredible lineup of speakers, both from India and US. We have patient advocates who have been involved in promoting research, and we are going to listen to their stories. And we also have some experts who are currently on our research core who would be talking about their research expertise and how they are trying to improve our knowledge about rare diseases. So first, we would like to launch a quick audience poll. Sadiq, if you can help us. So there are about 3 questions, and I'll give a minute. I'm not able to see the number of responses coming in, so maybe in another 10 seconds or so, you can stop. Can you close the poll? I'm not able to see. Yep. Okay. I hope you can see the results. So we have about 63% of the individuals are non-researchers. So about 40% are researchers. And they feel like they're probably least connected with IndoUS, with other researchers and patients and industry. So it's about equal. About 25% think they are okay connected, reasonably connected, but 50% thinks they are less connected. And they do think that they agree that we are able to help IndoUSrare as an organization. Are extremely, most of the majority of the people think that we are able to help with making those connections. Thank you for your opinion, we'll definitely work with you. Please find us and let us know how we can help to increase your collaborations with each other, with researchers and as well as physicians or pharma. So with that, I just wanted to start today's event. Few housekeeping events. Please type in your questions in the Q&A box that you see down at the base of your screen. A short feedback survey will be sent out after the session, and we request that you complete it promptly, and that's the only way we can improve. The event will be recorded and be available online to all who have registered. So even if you're not able to attend all the sessions you missed yesterday, please go back and look at those recordings, because yesterday's talks were incredible, and I will highly recommend you go back and listen to them. So with that, I would like to introduce our first speaker, Dr. K. Thangaraj, who is a colleague and friend. He is a director of Center for DNA Fingerprinting and Diagnostics at Hyderabad. Formerly, he was at the Center for Cellular and Molecular Biology as the chief scientist and a group lead. And he's been working on this area of genetics for several years. And his main area, I've always found fascinating to see his interest in the origin of humans, modern humans, and also the medical genetics, the genetics behind what causes diseases. And he's been working on cardiovascular diseases and rare mitochondrial disorders, as well as male infertility. And you can see that his productivity from his number of publications he has, and he has published in leading journals like Nature, Genetics, and Science from India, and has a big list of awards, and you can read them all here, and he has even a Lifetime Achievement Award. I know that he has much more to contribute, but I already gave him a Lifetime Achievement, and he's on board director and a board editor for several journals, as in leading journals again. So, and he's going to talk about his work on this rare mitochondrial diseases, as well as the overview about rare disease research in India. So with that, Dr. Thangaraj, please take it from here.
Thank you very much, Reena, for inviting me to talk and also giving introduction. It's my pleasure. Good morning to all of you, and good evening to all the colleagues joining from India. So I'll straight away go and share my slide. The slide visible? I could not see.
Yeah, we are not able to see. Okay. Sadiq, can you help?
Yeah, you are on the end of the last slide, Dr. Thangaraj. If you can click on the home button.
Sorry, 1 second. Oh, this is the first slide, right?
Yeah, I was able to read that it was the end of slideshow on that black screen.
Yeah, we are seeing it now. Maybe you want to do
it's not in full screen, right?
Mm-hmm.
Yeah, can you click on home button on your keyboard in that view?
1 second. Yeah, now it's okay.
Yeah, perfect.
Fine, right? Okay.
Yeah.
Yeah. So once again, thank you very much, Reena, and also Nisha, for inviting me to talk in this forum. It's my pleasure. And what I'm going to talk to you is mostly about Indian perspective. So why a large number of rare diseases in India? If you look at this slide, thanks to that Rare Disease India organization, that there are about 70 million people are suffering from rare genetic disorders. But if you see the number of every state, but that suggests that whether it's a rare or it's a common, right? So it's not really rare when you talk about any disease in India. When you talk about a rare disease, it may be a rare within India, but if you go to a particular population, it's highly prevalent. So what is the reason for that? And for that, we need to understand the Indian population structure. And it's not exaggeration to say that India is an ethnic museum, because we have a collection of large number of population groups. Some group typically look like Africans, some group typically look like Europeans, light skin color, some group looks like Southeast Asian. Some of them are typical to India. So India is actually having a huge diversity in terms of human population. And some of you might be wondering that is it true that India has more than 4,600 anthropologically well-established population groups that includes large number of tribes, primitive tribes, hunter-gatherers. And if you wanted to see the history of the Indian populations, and we all know that the Andaman tribes are the first modern human who migrated out of Africa and taking southern coastal route and reached Andaman and Nicobar Islands about 65,000 years back. So no wonder that why such a huge diversity exists. So not only that, and subsequent studies showed that the people who have migrated from Africa to India, and some group has stayed back in southern part of India, whom we call as ancestral South India. So in fact, this study where we used about 1 million single nucleotide polymorphic markers to understand the Indian population history, and where we have suggested that the Andamanese and Africans are the deep-rooted divergence, which we know about 65,000 years back. And I said that while Andamanese were migrating, some groups stayed in southern part of India, whom we call as ancestral South Indians. And there was second wave of migration. People from Africa moved towards north, and from there one group moved to Europe, and other group has come to northern part of India, whom we call as ancestral North Indians. So there are several evidence which I'm not showing because the time limit, to suggest that how we can say that there is a affinities between ancestral North Indians and Europeans. One is the mutation responsible for skin color. Another one is the Y chromosome haplogroup R1A1, which is very predominant in Europe, also predominant in northern part of India, but not in southern part of India. There are several other examples, so I'll not go into detail. And these two groups, ancestral North Indians and ancestral South Indians, have admixed and gave rise to many population groups. On the other hand, if you look at the PCA plot, these are all Indian populations. This is a European HapMap sample, African HapMap sample, and the Chinese HapMap sample. While you see that these three HapMap samples form a very tight cluster, look at Indians. Every single population is unique. That means they are following the endogamy marriage for a very long time. Therefore, they accumulated unique set of mutation that is very unique to that population. Therefore, that differentiate that from the other population. However, there's also a genetic affinities between each population. Otherwise, this cluster may not be like this. It may be clustering somewhere here and there. So every population is unique, but still they have a genetic affinities. So subsequently, to understand when this event took place, this admixture between ancestral South Indian and ancestral North Indians. So we took a large number of samples again from South and the North, try to understand that when this admixture event would have happened. Then we found that ancestral South Indian and ancestral North Indians initially gave rise to many population groups without any mixing of that. Then these two founding groups have admixed between the last 2,000 to 4,000 years. As this color indicates that the genome is between these two founding populations have admixed. After admixture, for the last nearly 2,000 years, every single population started maintaining the endogamy, means that marrying within the group. So most of you are aware that I mentioned that there are large number of population in India, particularly the caste population, tribal groups, and the caste system arose much later than the practice of endogamy, probably around 1,000 or 1,500 years. So that must have put more pressure on every single population started to maintain the endogamy. Okay, so this is the first part of my talk. Then the second part, what is the impact of the endogamy on the Indian population? To understand that, we took large number of samples, 2,800 samples from 275 distinct populations, this time we didn't take only samples from India, we also took sample from South Asia. The reason is that there are some mutations which is very predominantly present only in South Asian population. Like, for example, I did not have the slide. The mutation in myosin binding protein C3 gene, which causes cardiomyopathy, which is very predominant in India. Then we expanded the study all over the world, but we found that it is exclusively present only in Indian subcontinent. So that means that there are certain mutations which arose, that is around 33,000-year-old mutations. So that is much relevant, that whatever genetic change or the changes associated with the disease we are finding in India, that's also true for other South Asian countries. Therefore, we took the samples from other South Asian countries, including Pakistan, Nepal, Sri Lanka, and so on. What was interesting in this study was that we predicted that there's a possibility of identifying population-specific mutations which are responsible for population-specific diseases. And most importantly, the recessive diseases. Okay, recessive mutations and recessive diseases. So why we are saying that is that you can see this is the IBD score, identity by descent, and each line represents one population. And you can see most of the population in India have very high IBD score. And the length of the IBD score indicates there's a possibility of more relatedness among the individual. And when there is a more relatedness among the individual, then you expect that the mutation, particularly recessive mutations, are much highly prevalent in those populations. If it is a dominant, that is one chromosome carry the mutation, other chromosome is normal, then obviously you see that it is going to express the disease. But the problem with the homozygous mutation, the recessive mutation, is that it will not express any disease when it is existing in the heterozygous condition. Only when two individuals, the boy and girl who marries and who carry the mutation in heterozygous condition, that's a recessive mutation when they marry. So there's a possibility that 25% of possibility that they transmit the mutant chromosome to the next generation. Therefore, there's a disease. So that is very, very high among in Indian population, as suggested by this IBD score. So you also looking at here, there are two red color lines that indicates one is Ashkenazi Jewish, other one is Finnish population. You know that these two populations were well studied, and there are a large number of disease and disease causing mutations have been identified, and most of them are autosomal recessive. Okay, they also practice very high endogamy, these two populations. And look at the Finnish population. So there are also large number of disease and has been mapped. Okay, then imagine that what would be the consequences of large number of Indian population groups, because most of them are in the rural area, rural India, and they may not have opportunity to go to cities and show them that there's something disease existing in the family or in the population. Therefore, that one has to study very extensively in some of these populations to identify disease causing mutations. So to explain that in a simple term, that how it happens, that this is a pedigree, as most of you are aware, and look at here, there is a mutation in one of the chromosomes. It's a heterozygous condition and is being transmitted generation after generation. The father is without any mutation, therefore no problem in this generation. Again, both of them married somebody from outside, so the mutation goes generation after generation in a heterozygous condition. And after several generations, they might have lost their relationship. Okay, although they have come from a common ancestor. If you trace back, so you can go to a common ancestor. But if it goes for many generations, And that we generally see in a village, there are 100 families. Actually, the 100 must have come from a common ancestor. So if it is a recessive mutation going to increase its frequency in the population because they practice endogamy, and there is a chance that two male and female have the same mutation, and once they marry, they're going to transmit this one, both the alleles to the next generation. Therefore, there is a disease. So that is very, very high among Indian population groups. This is one example of one of the mitochondrial diseases. Polymerase gamma is a nuclear gene essential for mitochondrial DNA replication. You can see this is a wild type, and this is a mutant, and the heterozygous. Where you see that it's a consanguineous family where both the parents are heterozygous, absolutely fine. There's no problem at all, 45 years and 39 years. But unfortunately, they transmitted the abnormal chromosome to two of their three children, and therefore they have the disease, and which leads to mitochondrial DNA depletion, encephalopathy, seizures, stroke-like episode, and most importantly, the early death, premature death. It's a non-consanguineous family. Again, you can see that it's the same. So this is one example. This is another example where you can see this is one of the mitochondrial disease, mitochondrial neurogastrointestinal encephalopathy. Right? Again, you see that this is a wild type, complete mutant. The parents are heterozygous, and you can see that this lady has sort of multiple symptoms, because that's what the mitochondrial disease suggests. So we actually knock down the gene in zebrafish to see whether that mutation is really a cause for that. You can in fact compare the phenotype. This is a control where you see that it's perfect, and this is a mutant. So the knockdown one with the TIMP gene, and the phenotype is almost same as that, the loss of muscle and so on. This is another example of mitochondrial disease, C10ORF, which leads this mutation, leads to multiple mitochondrial DNA deletion, and you can see loss of hair and eyelash, poor and thickening of eyebrows, and so on. And most importantly, looks like premature aging. And he is actually only 22 years, but appears like 50 plus. Okay, so there are several such examples. Again, you can see that there's a multiple mitochondrial DNA deletion because of the mutation FOLG2. Again, homozygous mutation leads to mitochondrial diseases. As a mitochondrial disorder, I think there's one talk I have seen the slides. I'll not go into detail, but to show that even the number somebody sees, there are more than 1,000 mitochondrial disease patients which we analyze in a short period of time. And you can see that LH1, laboratory optic neuropathy, is 25% of the cases, which is supposed to be very rare. Similarly, chronic progressive external ophthalmoplegia and many more are quite common, and some are very, very rare. So what, in addition to doing research and diagnostics and providing reports, we also have a website. Somebody want to see, can go into that, and we created the website. We have the Society for Mitochondrial Research and Medicine to help the patient group with mitochondrial diseases. So now, based on the report which I mentioned earlier, that most of the Indian populations are with the high IBD score. So what we are doing right now is we identify the population which are with the high IBD score and pick up those population. We are sequencing 100 from each population. We also identified a regional population specific disease like Handikudu disease. This is from Karnataka. It's a place called Handikudu where people are very short stature and bone abnormality. That's exclusive present there. That's why the name is Handikudu disease. And this is Udanam kidney disease, exclusively only in the place called Udanam. Similarly, there are many more such diseases are existing, highly prevalent in the population, but very rare in Indian scenario or worldwide. So this is ongoing. And we are also planning to have a pediatric rare disease program. So where we are planning to have networking of at least 20 laboratory all over India, as you see here, then do the exome and whole genome sequencing, and use the tool to predict whether the mutations are really cause for that, then use different animal models to model the disease. Then we are also planning to do, of course, later on I'll come back to IndoUSrare. There is any opportunity for treating this patient, but initially we thought of using lentiviral mediated gene therapy, and we wanted to do a lot more awareness program for rare disease in India, particularly pediatric rare disease. So that's our future plan. Then I end with that by thanking all my colleagues from CCMB and also from CDFD. Thank you very much.
Thank you so much, Dr. Thankaraj. I was about to say that you have 1 minute left and you already did it on time. So thank you so much. And that was really very engaging. And I mean, I really did not know the history behind why we are seeing so much incidence of rare diseases in India. So it was really eye-opening and really educational. Thank you so much. We have one question from Ipsa Mahapatra. She's asked, what is the denominator in the prevalence of rare diseases slide you showed, the second slide?
So what is the question? Denominator of
the
number, that's actual number.
It's actual number?
Yeah.
Okay, no, I mean, but which number? The prevalence, you were showing the prevalence data in the second slide, I think that's what she is asking.
Yeah, so that's in each state.
The actual number.
Actual number, yeah.
Okay. So it's not a proportion. Okay, got it.
No, it's not proportion.
Okay. Any other questions? Let me check the chat box if anybody put in a question. I don't see any. Maybe I just had
very underestimated number, because you know that we don't have a clear epidemiological data to have exact number, because mostly these are all hospital-based data. So if you go to rural, you might find much more. And the information from rural are not registered elsewhere. There is somebody who dies at very early age, for example. So there's no account for that. And they just ignore that, okay, it might have happened something.
And they may not get diagnosed.
Yes.
Yeah. And I just had another question. Yesterday, Dr. Kabra was talking about Center for Excellence, and I thought there was one Center for Excellence under the new National Rare Disease Policy. And there is one Center for Excellence, I think, in somewhere Hyderabad or in and around Hyderabad.
There are 10 centers. So they wanted to start treating the rare diseases. The Ministry of Family Welfare is very much determined to do that. In fact, CDFD is one of the center. AIMS is leading that program. AIMS can do directly the treatment part, but we cannot do directly. So we are in collaboration with NIMS, Nizam Institute of Medical Sciences, because ours is not the hospital. So
but the center also contributes for research.
Yes.
And hopefully there will be options for collaborations, and we are looking forward to seeing how we can help through our research core and also our patient alliance members.
Sure.
Thank you so much. Is there one more question? Okay, is there any surveillance program for rare diseases in India?
Surveillance, once you start a program like this, or Dr. Madhulika said, and also I think Vinod would be talking more. So we'll get more information only by doing this. Yeah,
good. Thank you so much, Dr. Thankaraj. Please stay on. We have a networking session, and hopefully there will be more questions that would be coming in at that time. So let me share my screen. So now I'd like to introduce our second speaker for today. And that is Ms. Sophia Zilber, who is a board member and patient registry director at Cure Mito Foundation. So we will be continuing our discussions around mitochondrial diseases this morning. So Sophia has been working on the pharmaceutical industry, focusing on data analysis and statistical programming of clinical trials data. And she was introduced she pivoted into the rare disease field after she lost a child to a mitochondrial condition, Leigh syndrome. And following that, she has been volunteering a lot of her time to help the rare disease community, especially to increase raising awareness about regarding patient registries and what is involved in collecting high quality data. And today she's going to share her experience and her journey, as well as probably the research that has been taking along on the site. Sophia, please take it from here.
Thank you. I'm going to share my screen. Yeah, please let me know if you can see my screen. 1 second.
Yep, we can.
Yeah, thank you so much. Thank you for having me here. So my name is Sophia Zilber, and I'm a board member and the patient registry director for CureMyDA Foundation. Professionally, I work in Pfizer in statistical programming and analysis in rare disease. And my personal connection to rare disease, I unfortunately lost my daughter Miriam to Lee syndrome, which is a type of mitochondrial disease, several years ago. A couple of words about CureMyDA Foundation. CureMyDA is a parent-led nonprofit organization. It's dedicated to advancing education and research for Lee syndrome. It's a fully volunteer organization. All funds we raise are going towards research. CureMyDA was actually founded in 2018, originally as CureSurf1 Foundation. There's been a lot of work done to fund preclinical research of SURF1 Lee syndrome, which now is hopefully on the way to have gene therapy clinical trial later in 2022. And just recently, a few months ago, CureSurf1 expanded its mission to all of Lee syndrome, all mutations, and changed the name to CureMyDA. So what is Lee syndrome? It's a type of mitochondrial disease. Symptoms typically appear very early in infancy or early childhood. It affects 1 in 77,000 births. It's a fatal disease, and few kids live past 10 years old. It could be caused by over 100 mutations, either in nuclear or mitochondrial DNA. I'm going to talk about our patient registry and some of our other projects on data. So currently we have 3 projects related to data. One is our worldwide patient registry for Lee syndrome. We are doing it in collaboration with CORDS, Coordination of Rare Diseases at Stanford. We also have medical record collections for Lee syndrome with all stripes. And we have a new data sharing collaboration with Critical Path Institute. So first, about our worldwide patient registry for Lee syndrome. CORDS is a platform, it's a disease agnostic platform. Currently, it has over 1,800 diseases on it, data for over 1,800 diseases. There are 83 partner groups, over 13,000 participants, and all 50 states and 79 countries are on this platform right now. The way this platform is set up, there are 2 surveys. One, one is a general, one survey is a general survey. General survey in CORD is exactly the same for all rare diseases on that platform. It's based on the survey that was developed by NIH Rare Diseases Office. And because the survey is the same for all rare diseases, it makes it easy to harmonize data across diseases and look for similarities and things like that. Additionally, there is a disease-specific survey that's developed separately for each disease. So since there are a lot of mitochondrial disorders, there is a potential if we add other minor conditions, general survey would remain the same and a disease-specific survey would be different. So it would be easy to harmonize that data and see what we can see in common, using the general survey. And our Lee syndrome survey could potentially be used as a basis for other mito disorders. CORDS is linked to Orphanet, a list of rare diseases. So when patient signs up, the rare disease in the list, it's not determined by any patient group, it's not determined by codes, it's automatically linked to Orphanet. And the patient can join even if there's no advocacy group represented. So the joining into this platform is based on disease. It's not based on a patient group. Multiple family members with the same disease can be linked together. A few more features of CORDS. It's an IRB approved protocol. This data is protected by HIPAA because Stanford Health is a healthcare organization, and so they're considered a covered entity under HIPAA. It's GDPR compliant. There is no cost to patient groups or for researchers to access data. Everything is funded by Stanford Health. If a researcher wants to request data, or if a company wants to share a clinical trial opportunity with patients, they just make a request through a researcher portal. It's a very transparent and a very easy process. Each participant has a global unique identifier, GUID, and so it's possible to link these patients with the same patients in other registries or in other data sources, and that can be really valuable for rare disease data. A few words about our Lee syndrome survey. It was designed with the input and review from our registry advisors from the industry and with review of our scientific and medical advisory board. Some of the information we're collecting is diagnosis and genetic mutation information, symptoms and concerns that were first noticed, loss of milestones when a child lost milestones, what is used for disease management, symptoms experienced, healthcare utilization such as number of hospitalizations, emergency room visits, hospital admissions, and also caregiver burden information. So this is the current status of our registry. We just opened it exactly 2 months ago in September. And now we have over 120 participants enrolled and more than 90 who responded to surveys. So this is really, really amazing result for just 2 months of our registry. And based on the data analysis so far, we have 21 countries now in the registry. Russia is the country that has the most participants now. So it's interesting because we're a US-based registry, but looks like we are really international. 92% of participants are living. A diagnosis was determined by genetic testing for 96%, and out of those, for 92%, we know their genetic mutation. So that's very, very good. Our data looks very good and looks like it's very useful, and we're working on data analysis and planning to report detailed results very soon and share with everybody. So our vision for this registry is to really know our patient population everywhere in the world, who our patients are and where they are. We are very committed to transparency. Any questions are welcome, any feedback is welcome. We are open to collaborating with everyone. We really want this to be a community effort and be transparent with everybody. We are going to share findings from this registry with, again, with medical community, with patient community on a regular basis. We want everyone to know what's in the registry and for patients to know that they are participating and they know what the findings are and that their participation really matters to this. We will be sharing clinical trials and research opportunities with patients through the registry. And we hope to also just build a stronger community, patient and medical community, and empower patients with this registry. So now I will say a few words about our medical records program with All Stripes. We are collaborating with All Stripes. MITA Action is another MITA group. They're also a partner on this. All Stripes collects medical records on behalf of patients. So the way it works, the patients give their consent, and then All Stripes will obtain their medical record and will digitize them and will structure them into data sets and then this data could be shared with researchers. IndoUSrare updates the information once a year, so the information can be tracked over time. And information can be shared with researchers, and patients too can see all of their medical records organized together in one place and share with doctors or use it for any reason. We are hoping this will be helpful. This is very different from some of the platforms where patients themselves upload their electronic health records, because when patients upload their records themselves, it doesn't necessarily give a complete picture of all of their medical records, because patients obviously can't. It's a very, it's a huge burden for patients to obtain all of their records from different medical providers and upload them. And after that, what happens? Records are usually in the PDF files, so they have to be digitized. So here, this is all done by IndoUSrare. And additionally, this is done every year, so it's a longitudinal data in the end. And the countries now eligible are US, UK, and Canada, but we are hoping that more countries will be added soon. And
1 minute, Sophia.
Yeah.
You have 1 minute.
Oh, I'm sorry. Yeah, this is my last slide. We recently started a collaboration with Critical Path Institute. They have a platform on RDCA there. It's a FDA-funded platform where different data sets can be integrated and more can be learned from the combined data. So here I'm just sharing our press release on this. There's more information on our website. This is not just for Lee syndrome data, this is for any mitochondrial disease data, and we welcome conversations with anyone and any collaborations and further conversation on this. Yeah, this is my last slide. Please feel free to reach out with any questions. We are very happy to work with others. Yeah, thank you very much for having me here.
Sure. Thank you so much, Sophia. Thank you for sticking to the time. I just had a question. So you were talking about the results would be published. Do you know the timeline? When do you think that would happen?
Yeah, we are planning to share the results in February.
Okay, great.
February,
okay.
Yeah, and I can connect with you to give you more details, but in February, that's the plan.
Good. Yeah. And I'm sure people who are interested in participating will know how to find you, because we'll be posting these recordings and they can always hear more about the registry, like go back and get the information they need.
Yeah. And even before we publish the results, if anyone has questions, we already have this data in good shape. So we're always happy to discuss and share.
Sure. Okay. Thank you so much, Sophia, for coming and joining us today and for giving us this information. Thank you.
Thank you for having me.
Mm-hmm. Okay, so let's move on, and I'm going to share my screen, and I'm going to invite our next speaker, Dr. Vinod Skaria, who is a clinician turned computational biologist. His research interests are in understanding the function, organization, and regulation of vertebrate genomes and how the variations can impact them. And he's the co-founder of the Genomics for Understanding Rare Diseases India Alliance Network, or the Guardian program. It's one of the largest network of clinicians and researchers in India working on rare genetic diseases. And this research has contributed to several new technologies and insights that has led to improvement in diagnosis and screening for rare genetic diseases. He has also have a lot of publications and a lot of awards under his belt. And today he's going to talk about his contribution to the rare disease area. Vinod, please take it from here.
Thank you very much for the nice introduction. Let me please share my slides. Yeah, thank you for the nice invitation and for the great introduction. For the next 20 minutes or so, I will give a brief overview of our journey in the area of rare genetic diseases in specific and population genomics in a whole. Before that, let me make a few mandatory disclosures, because we work with a number of companies, both nationally as well as internationally, in the area of rare genetic diseases. And we receive personal as well as research grants from many of these companies. Also, just a slide on a brief overview of what we work on. We work on a variety of areas in and around genomics and informatics. right from looking at Mendelian genetic disorders, trying to look at the genetic epidemiology of diseases from population scale genomes out of Asia, Middle East, and Africa. And of course, a bit into looking at pharmacogenomics and pathogenomics. I'll not delve deeper into that in this particular talk. So the talk today is a bit of the journey for almost over a decade. We are fortunate to have been in this field and fortunate to be in the right time, probably. Almost a decade ago, we sequenced the first genome from India. And the idea of sequencing this genome was to actually learn the trick of the trade, not just to sequence a genome, but also to put the analysis in place through a variety of computational analysis and to give a clinical perspective to genome analysis in terms of what is actionable if you do a personal genome. Now, quickly moving forward over the last decade, we participated quite actively in national as well as international programs on genome scale sequencing in this region of Asia and Middle East. We're one of the founding members of the Hugo Pan Asian Population Genomics Initiative. We're fortunate to have collaborated quite extensively to also in the programs to sequence and analyze the Malaysian, the Sri Lankan genomes, and we also have contributed to extensive analysis of the Middle Eastern genomes. And today we hold the largest databases of genomics, genome sequences from this part of the world, which is not quite represented in global sequencing expeditions. This includes the SAGE for South Asian genomes, Indigen for the Indian genomes, and Almina for the Middle Eastern genomes. All these are public resources. Now, one of the applications of doing such large-scale genomes is to understand the underpinnings of genetic diseases. And to put a number to this diseases, I'm sure you've heard this over and over again. There are around 70 million people in India suffering from one or the other genetic disease or trait. This in many ways could just be an underestimate. And in India, many of these patients go through multiple rounds of clinician visits, multiple rounds of misdiagnosis, and many years lost before they can even get to a diagnosis. And that's largely because there's a lack of awareness, even among specialists on genetic conditions. There's a lack of mutational understanding on the mutation spectrum of many of these genetic conditions. And there has been no comprehensive system for diagnosis or screening of many of these conditions. To start from scratch, we put together a program called the Guardian. Guardian stands for the Genomics for Understanding Rare Diseases India Alliance Network. It starts with a clinician who sees an abnormal case or a family of probable genetic condition, who refers to a program where we do a systematic analysis of the genome after consenting. And when we return a result, which is possible in approximately around 30 to 50%, depending upon the specialty, the clinician can take an action upon that report. But as I said, the rest of the cases are still unsolved. That is what we take up for further in-depth analysis using in silico, in vivo, or in vitro systems in an assumption that we'll be able to return back the pathogenicity of a variant at some point of time. And today we have grown over 300 collaborators from around 70 centers across the country, spanning the length and breadth of the country, to also make it one of the largest networks for rare genetic diseases in this part of the world. And this is not just a program for looking at genetic diseases, but to actually take up right from the perspective of advancing research, advancing education, advancing diagnosis, and also contributing to policy and implementation of policy. And to know more about this program, you could actually look at an overview which we published in 2019. And the core to this program is essentially data analytics. And thanks to the genomic data that is widely available and can now be sequenced and accessed, we have a quite elaborate computational system which indexes many of these genomic data sets and also put it into the perspective of genetic diseases and pathogenicity of genetic variations, or in other words, looking at evidence from others' studies as well as looking for evidence in genomic studies. Now, I'll give a few examples, not many, just to give a brief perspective of what really happens in this program. Just a few examples so that you can get an idea. And this is one of such cases which were referred to us sometime in 2015, because this family actually asked the President of India permission to euthanize all their 5 kids, rather 6 kids. And that was practically because all the 6 cats had a progressive neurological disorder without any diagnosis. And nevertheless, there were multiple provisional diagnoses with no molecular confirmation. So right from 2016, where we could access this family, we went back and studied quite extensively this particular family and this community. And just by a survey, we could identify additional families. Just by doing an extended pedigree, we identified 4 families with a similar condition running in a quite consanguineous group. By genomic studies, we did identify this particular genetic disease as MLC1 or megalencephalic leukoencephalopathy with subcortical cyst type 1, an extremely rare genetic condition. And we could test a large number of individuals, and we could identify additional new individuals in this particular community for this extremely rare genetic disease and an extremely rare genetic variant. And from there, we could establish frequency of this particular genetic mutation in the population to be around 28%. Since this is a quite large community spread between multiple states in the country, we could roll out education and awareness and actually put out a genetic test, which is today available in one of the major path laboratories in the country. So from 2016 to 2019, you can not just solve the diagnosis of the disease, which is essentially a big diagnostic OTC for the family. We could estimate the carrier frequency for this genetic disease, otherwise extremely rare in global populations, but quite prevalent in the small endogamous group. We could publish this, and of course, you could go back and put the diagnostics in the market. Now, as clinicians, we all tend to believe that we do see genetic conditions in the clinic, but that is essentially a myth. And that's practically because many of these genetic conditions do not really come to the clinic because they do not have a deadly manifestation or a very severe manifestation. One of such example is this hypogonadotropic hypogonadism. Apart from the fact that they have small gonads and they have lost their reproductive function, they can otherwise have a perfectly normal life, and they might actually not really come to the clinic. And therefore, practically all estimates of genetic diseases from a clinical setting could actually be error prone. Now, how do we solve this problem? The problem can be solved only by taking representative samples of the population of normal individuals and ask this question: what are the carrier frequencies for pathogenic genetic mutations in the population? And we did that quite recently. Along with Dr. Tangraj, we did this program called Indigen, which is a pilot program to sequence 1,000 odd individuals from multiple populations in the country. And one such analysis I could explain in the next couple of slides. We looked at treatable causes of intellectual disability. There are 116 disorders with around 139 genes, which are involved in intellectual disability, but nevertheless is a treatable cause of intellectual disability. The advantage for such causes is essentially that you have a very specific therapy or management option presently available anywhere around the globe. The only limitation is, of course, you need to look for evidence, and you need to sift through thousands of publications to actually look for this evidence and put them in the context of the ACMG and the AMP guidelines in terms of pathogenicity annotations. We did that quite elaborate job over a year and a half, and we identified a large number of pathogenic and likely pathogenic genetic variants. Now we could go back to the population data and ask this question: how many of them are quite frequent in the population in terms of carrier frequencies? And what we found was quite surprising, that there are a large number of diseases which seemingly are underrepresented in the population. For example, if you look at Wilson's disease, has a carrier frequency of approximately 1 in 53 in the Indian population. Phenylketonuria has a carrier frequency of 1 in 129. Citrullinemia has a carrier frequency of 5 in 1000, so on and so forth. Now, the real question here is then, why aren't we seeing such patients in the clinic? And the quick answer is many of such patients do come to clinic, but they come to very different kinds of specialties with very different kinds of clinical manifestations. So we really rolled out a program, and this was led by Dr. Binu Kumar, who is my clinical colleague and collaborator. He rolled out this program spanning multiple states in the country, putting together clinical collaborators and educating them on how to really look for Wilson's disease. And over the last 3 years, we have actually created one of the largest cohorts for Wilson's disease by just educating clinicians and asking them to look for very specific signs and symptoms and lab investigations so that we can actually build upon the cohort of Wilson's disease. So in other words, what I'm trying to say here is that there are genetic diseases which are underrepresented in clinical settings just because clinicians are not able to identify them. There are no clinical tests that are available, and there's no molecular confirmation which is available. And that's what we want to change. So we rolled out a very specific hotspot panel, which is accessible and affordable, so that a large number of these patients could access genetic test. Going forward, we could also identify genetic diseases which were never found to be prevalent previously in the country. One such example is immunodeficiency disorders or primary immunodeficiency disorders. The good part of such immunodeficiency disorders is that early diagnosis, early in the childhood or neonatal stage, could actually save the children because they have definitive therapies available, either intravenous immunoglobulins or by bone marrow transplantation. And just from one center in South India, which also happens to be my alma mater, we could identify a very large cohort of 350 children. A significant number of these children could access very definitive therapy, either IVIG or bone marrow transplants. And right from this diagnosis, we took them up to a specialty clinic, which was established locally in the hospital. We offer diagnostic support in association with the state government, as well as philanthropic organizations, and a treatment plan put in place, also funded by the state government, so that many of these children could be diagnosed very early, could be accessing very definitive treatments so that they are alive. And of course, going further, many of these families would require genetic counseling and prenatal services, which is also set up in the center. And going forward into the future, and the future is essentially digital, what is really important is to have a system to communicate genomic evidence, which also is a very dynamic thing altogether. And this is very specific because of the fact that evidence changes and evidence improves at every passing date. And therefore, we need to incorporate this ever-changing evidence into the genomic context in terms of interpretation. And 2019, we launched this program to actually communicate digital clinical information, or rather genetic information, based on the standard guidelines of ACMG AMP pathogenicity into an app and a card. This was launched in 2019, which provides a secure, privacy protected, scalable point of care, and more importantly, clinician enabled access to genomic information and interpretation. So in summary, what we try to build is clinical programs where we work with clinicians to educate them and make them aware about genetic diseases. We also offer genomic testing services. This was run by my colleague, Dr. Mohammed Farooq, today offering around 130 high-end genetic tests as part of a program called GoMed. We work with the industry, the diagnostic industry, to roll out affordable and accessible genetic tests, in many cases, very specific genetic tests for prevalent genetic mutations in the country. We work on research and drug discovery, both using in silico as well as in vitro and in vivo tools, including model systems like zebrafish. And of course, we go back to the population, trying to figure out unknown and undetermined genetic diseases by looking at population scale genomes to understand the frequency for such diseases in very specific pockets around the country. In summary, what we try to understand is to put genomics in the perspective of 5 Ps, making it predictive, making diseases preventive, making diagnosis very precise, and treatments very personalized. But the central P is the most important of this all, which is the participation, not just of patients and families, not just of the caregivers, but also a whole gamut of ecosystem around, which includes the patient support groups and the civil community. I think I'll stop there and take questions, but before that, let me acknowledge a lot of people who made this possible. Fantastic collaborators within IGIB, Dr. Sridhar, who was a co-founder of Guardian program, a fantastic set of clinical collaborators spanning length and breadth of the country, and of course, consistent funding of the last 1 decade from CSIR to multiple different programs. Thank you very much.
Thank you, Dr. Scaria. Sorry, my mute button was not working, so I got a little glitch there. That was really an interesting presentation, and I mean, the amount of work that is going behind the scene is something which you don't really realize. I mean, number of people and the amount of work that is going behind there. So I just was really intrigued by when you said that, I mean, most of your databases are publicly available. So when we conducted our feasibility studies for patient-reported data and registries, there was skepticism about data sharing. And maybe people are concerned about misuse of data. So how do you win the trust of your patients, and how do you go about getting their consent?
Yeah, so when I said the data sets are available, that doesn't mean that the individual level data sets are available. These data sets are essentially summary level data sets for which is useful for clinicians and other researchers. So individual level data sets, we don't really share because there are many issues. For example, we don't have a Non-Discrimination Act in place. In India, you could get discriminated if your genomic information is available. So there are many other issues that need to be solved before this genomic data can be made available public, like the rest of the world. But until then, the personal information and the personal genomes are not yet available. But the summary level information is really available.
Okay, good. Thank you so much. Dr. Anahita Hegde, who is one of our panelists today, she has a question. As a clinician, I was not aware of the great work you all do. Is there a way to promote and reach out to clinicians?
Yeah, so we do a variety of programs to educate clinicians, which includes a very recent program, thanks to the pandemic, which became online, which is called the Genomic Variant Analysis and Interpretation course. On an annual basis, we train close to over 1,000 clinicians, both within the country and across the globe, in our ways of interpreting clinical information and pathogenicity of variations in a very systematic way, conforming to the ACMG and MTP guidelines.
Okay.
We do also conduct CME programs for very specific diseases where we want to, for example, Wilson's disease. We have a very specific focus, and we do have very specific CME programs for those diseases.
So I just had a question about the Wilson disease program. I mean, you had said put in a list of centers where you are in launching it. Is there any criteria how you choose that? Is it because of, that in that area there is an increase, there would be an increased risk, genetic risk or something?
Yeah, so both of it. It starts with our understanding about the increased risk or the chance of finding a Wilson's disease patient. And second, of course, a good collaborator who's also equally committed and interested in looking at a particular disease. So it's a mix of both. Okay.
And I know that there are a few researchers in the US who had reached out asking about how can they get a particular gene of interest on a diagnostic panel. Do you have any recommendation or any advice for them? Actually, our next speaker is going to talk about that. And I think she just put a comment here on the chat.
I didn't really get the question.
Sorry. Yeah, so I mean, there are individuals, like our next speaker is going to talk about a gene that she discovered, where there is a disease associated with it, and they're trying to figure out how they could, and that may be a scenario where they would like to include that gene in the diagnostic panel. So you are talking about one of the programs where you have like a medical genetic panel.
Yeah, so typically for the Guardian program, we use either a whole exome or a whole genome sequencing, because we typically take up undiagnosed genetic conditions. But for prevalent genetic diseases, which we have very specific cohorts, like for example, primary immunodeficiency or Wilson's disease, right? We do create very specific panels. And yes, if the disease is prevalent in the population, we could actually add the genes and mutations into that panel. We are very open to doing that.
Okay, and again, I mean, this presentation will be available online, and so for researchers and experts who are trying to reach out, probably contacting you would be the best way to get into those collaborations and discussions. But we are always there to help and act as the conduit, trying to link the collaboration. So thank you again, Dr. Scaria, for taking time to join us today. I know that you're some extremely busy person, so we really appreciate you being here. Thank you so much.
Thank you very much. It's my pleasure.
Good luck with all your work.
Thank you very much.
So let's move on, and I'd like to share my screen again. Sorry, excuse me. Okay, so now I would like to invite Dr. Julie Saba, who is a professor at the University of California, San Francisco. She is a pediatric oncologist, and she also holds the John and Edna Beck Chair of Cancer Research. So again, she has a medical degree from Johns Hopkins and a PhD in cell biology from Duke University. And during her research, she identified a gene. This was again serendipity when they working behind the screen. She identified a gene which was the first sphingosine phosphate lyase gene. And with several years of research, she also identified a disease that is associated with this gene. And that's what she's going to talk about today, her journey and where she is with her understanding this particular disease as well as the gene. So Julie, please take it from here.
Okay, I'm going to share my screen. And just one moment. Okay, can you see my screen all right?
Yes, we do.
Okay, great. So first of all, I want to thank the organizers so much for inviting me to this meeting. It's very exciting, and I am already popped questions into the chat box to try to connect to some of these wonderful speakers. I am going to be talking as a researcher. I'm not a patient or a patient advocate, but I'm doing my very best to advocate for children and families dealing with SPLIS, which is a newly recognized inborn error of sphingolipid metabolism caused by mutations in the SGPL1 gene. This is just a picture of two of the patients with this very ultra rare disease that we are still trying to determine the prevalence of. So this just is a little snippet from the patient advocacy website that we are actually developing. It tells you what SPLIS is. It's a rare genetic disease in which a type of lipids called sphingolipids that are abundant in all the cells of the body build up because of a failure to metabolize them, causing damage to the kidney, the brain, adrenal glands, and other organs. It's a recessive disorder. So children have to have a mutant copy of the gene from both mother and father in order to be affected. The syndrome has many different features. One is lymphopenia, which is a reduction in the number of circulating, mostly T lymphocytes that are involved in immune function. And so the patients can be susceptible to infections and have an immunodeficiency. But the most serious feature of the disease that brings the kids mostly to medical attention is something called steroid-resistant nephrotic syndrome, which affects the ability of the kidney to filter the blood, which is one of the kidney's main functions. And when the kidney is not able to do that, very important proteins, such as our antibodies, clotting factors, albumin, all these very important large molecular weight proteins get spilled into the urine inappropriately. And this form of kidney disease in this group of patients progresses very rapidly to end-stage renal disease, and the kids will not survive without peritoneal dialysis or transplantation or hemodialysis. Many of the patients also have adrenal insufficiency, so they can't make cortisol, our stress hormone. Some of the little boys can't make enough testosterone to have a normal genitalia. So you can see hypogonadism in the males. And then there are neurologic defects that generally manifest as a peripheral neuropathy with numbness, tingling, pain in the hands and feet, loss of muscle strength. And some of the patients have a progressive neuropathy that can lead to death. The infant children with the disease have a pretty high mortality in the first few years. And as this graph shows, the earlier the patients are diagnosed, the more likely they are to die of the disease within a very short time after diagnosis. The cause of the disease is mutations in the SGPL1 gene. This is a gene I first discovered in budding yeast when I was looking for cancer genes 25 years ago, and as a pediatric oncologist. And so I'm very knowledgeable about this gene and the enzyme that it encodes. And that's why I'm so focused on trying to use my expertise to help these families. So the function of the enzyme is to break down a molecule called S1P, sphingosine phosphate, or S1P for short. And so the enzyme is called S1P lyase, or SPL. And the S1P is an important signaling molecule involved in immune cell lymphocyte trafficking and angiogenesis, and that may underlie the reason for the disease. Importantly, this enzyme requires vitamin B6 to work, and that will become very important later. So this is a complicated slide, but I don't want to go through the whole metabolic pathway, but only to mention that SPL is at the very bottom of this pathway. And it's the only exit point for sphingolipid metabolism. So when this enzyme is not working, not only S1P, the enzyme's substrate, but other sphingolipids upstream accumulate. There's a complete blockage, and some of these lipids like ceramide and sphingosine can cause cell death, and that probably contributes to some of the manifestations of this disease. If you look at the cells, in this case, we're looking at skin fibroblasts from a punch skin biopsy from a number of different patients. And we look at for the enzyme itself. In most cases, you can't see it. So all the way on the left, we see a bright yellow signal, which is indicating that the enzyme is present in the cells and has a nice dispersed kind of pattern of expression. In all 3 patient samples, to the right, all you see is green, and that's where the lyase is supposed to be, where the enzyme is supposed to be, and there's nothing there. If you measure the enzyme activity in the patient fibroblasts, there's very little. And if you measure the S1P, which is what the enzyme is supposed to be breaking down, it accumulates. So this is a kind of a confirmation diagnostic test that we can perform on the patients. So far, there are about 60 patients in the whole world with this disease. It was only discovered in 2017 when we were performing whole exon sequencing of children with rare kidney disease and found several pedigrees. And since then, about 8 different research groups have come forth with patients' case reports. And although there are many different mutations that can lead to the disease, they tend to cluster right around the center of the protein. This is how the protein is encoded, and that is the cofactor binding domain. So that's where the vitamin B6 will bind to the protein. So not surprisingly, this is the most conserved area of the protein and where we see most of the mutations. So as I mentioned, because I've been involved in this, studying this gene and this enzyme for years and years, I figured if I don't find a cure for these kids, I don't know who will. And so I've been really focused on not just trying to understand the pathophysiology of the disease, but how to cure it. And I have taken 2 different approaches. One of them is related to vitamin B6, the cofactor of the enzyme, and the other is gene therapy. And I'm just going to tell you quickly about where we are with both of these. So vitamin B6 is an enzyme that's required for over 100 enzymatic reactions. It's a cofactor for about 100 different enzymatic reactions. And it's been shown that in a number of other inborn errors of metabolism that involve B6 dependent enzymes, that if you add high dose vitamin B6 to the patients, you give them supplemental vitamin B6 at high dose, that you can actually help protect the mutant protein from degradation and help it function better. It can have both a kinetic catalytic activity, but it can also function as what we call a chaperone, meaning that it can wrap around the protein and sort of protect it and help it to find its proper formation, its proper 3-dimensional or quaternary structural form, and that makes it work better. So we theorized that maybe that would work in the SPLIS patients. And to start out, we tested this in patient fibroblasts. And this is a study from one patient's fibroblasts where we're measuring cellular S1P on the left. That's a measure of how well the enzyme works. When it's not working, the S1P level is high, as you can see in the vehicle control on the left. And when we add increasing amounts of vitamin B6 to the medium where we're growing the cells, you can see the S1P level goes down. And that's an indication that the enzyme is working better. And if we actually measure the enzyme activity in those cells, we see that, in fact, vitamin B6 is making the activity improved. So that was exciting. So then we treated 4 patients with different mutations with vitamin B6, high dose vitamin B6, which is used very safely in infants, pregnant mothers, nursing mothers. It's a very safe vitamin to use at high dose. And we just simply looked at the lymphocytes in the blood of 4 of the patients. And I'm just showing you from 2 patients who responded that their low lymphocyte counts shown before treatment rose significantly after treatment. And this is not, making them all that much better. And these patients already had kidney failure, so we couldn't correct their kidney failure. But it did suggest that the patients might actually respond. And if we could get them vitamin B6 before they go into kidney failure, we might be able to protect them from both loss of kidney function, adrenal function, and the other problems in this disease. To the right, you just see the different T cell subsets, which are just different components of the immune system, which are also improved with treatment. And in one of the patients, we saw that the very high level of S1P in the blood, which is an indication of the disease, began to come down over the months after vitamin B6 treatment. So we have another 4 patients that are being treated now. We're trying to understand what the which specific mutations might be B6 sensitive, and whether we can actually protect the patients before treating them. And we have a mouse model that we are studying this in as well. So moving on to gene therapy, because vitamin B6 will only work in patients who are making a mutant protein, but there are many mutations that result in absolutely no protein being made at all. Those are the most severely affected patients. And for that, we need to do a gene or enzyme replacement. And enzyme replacement is complicated for reasons I don't have time to talk about, but gene therapy will address the root cause of the disease. And we know that we only need to get about 10% of normal enzyme activity to make these patients very likely better. And we're using something called AAV, adeno-associated virus, which is one of the most well-studied vectors for delivering a healthy gene into a patient with an inborn error. We're lucky that there is a knockout mouse model of this disease, and this is just showing you the survival curve and the size of these mice. They're very, very tiny. They die by 50 days of age. Usually they die at the time of weaning, which is at 3 weeks of life. And that will become important in a minute. And they have a lot of the same features. That's the disease. They have kidney disease. They have lymphopenia. They have all the same, really the same manifestations. And they also you can see the kidney defect here is shown on the left by urine ACR, which is a measure of the amount of protein that is spilled into the urine. It stands for albumin creatinine ratio. And it's very, very high in the knockout mice. They're inappropriately spilling protein. And when you look on the right, this is a biopsy of their kidneys. And on the top, you see a normal looking glomerulus, which is the part of the kidney that filters the blood. And in the bottom, what you see is an enlarged glomerulus that has a lot of pink tangles in it. And that's scar tissue. And that's why these kidneys fail, because they just develop scarring. So we have this knockout mouse. We cross heterozygote by heterozygote parents, and we get 25% of the pups have the disease. We genotype them. We do the genetic test at day of life 1, and they're pink when they're born, and you can see the facial vein. When we get a knockout mouse that has the disease, we inject them with a small amount of virus into their facial vein. We add a little bit of food coloring so the whole mouse turns green, so we can be sure that we've gotten the virus in, and then they get rid of that. They pee out the green food coloring in about 24 hours. It's very safe. And then we look at them for all kinds of endpoints. This is a biopsy of the brains of wild type mice in the top left, just showing you these brown cells that are stained for the enzyme. The knockout mice to the right have no signal at all. There's no brown because there's no enzyme. And the 2 squares on the bottom are 2 different virus treatments showing you that we have restored the enzyme to the brain. We also are able to correct the high levels of sphingolipids in both the tissues and the plasma of the mice. You can see the high levels in the knockout in the middle of each of these graphs, and then the AAV gene therapy treated mice are shown to the right, and you can see it goes down. But most importantly, we have prolonged the survival of these mice to almost a year with a single treatment. This is our very first treatment. Just 10 mice were treated, and we're already getting dramatic improvement. We're very excited about this. It's not perfect, but it's given us the momentum to move forward. Just to tell you that the kidney function is protected in the long-lived mice. Their proteinuria is completely normalized. Their blood albumin levels and the tuft area, which is a size, kind of a way of measuring the size of the kidney glomerulus. And you can see below that the knockout tissue is very large. The glomerulus is large and normalized by treatment. And lastly, neurologic function in these knockout mice are abnormal. The little newborn mice, less than 3 weeks of age, don't do very much, but they open their eyes on a certain day. They can hear, they can grip, they can hold on to something, and they can develop a normal walking pattern with their feet. And the knockout mice are delayed in achieving these things, and they're weak. But when we treat them with the gene therapy, they're completely corrected. So we're very excited about what progress we've made in just 4 short years. We are looking for SPLIS patients, and I'm very glad to have the opportunity to connect with many of you in the audience to try to see if we can help determine the prevalence, which is an ongoing project that we have with 2 genetics collaborators. And to do that in India would be fantastic. We know there are patients in India from our discussions with some of the genetic diagnostic companies, but they won't tell us. So we need to find other ways to find them and to find the alleles. We're very lucky that we have benefited from our collaboration with Jiva Informatics to be set up to have the first SPLIS patient registry. And we have a natural history observational study available on clinical trials. And we're just about to put up our patient support group website. So with that, I will end and thank you all once again and ask if you have any questions.
Thank you, Dr. Saba, for a very, again, informative presentation. And I think it's such a rewarding journey for you to have discovered the gene and find out the cause for it and understood the gene, identified the disease that is being caused by it, and trying to come up with a therapy. Wow, that is something, an achievement for your whole career, I guess. I mean, it's really remarkable. So there's one question here, and it's about, are ketones still being produced in patients with SPLIS, SPLIS?
As far as we know, they are making ketones. Is that the question?
Yeah, ketones.
Yeah, they make ketones. They're not ketotic though. They don't have an increased amount of ketones. Their main metabolic problem is they can be hypoglycemic. Like children with Addison's disease who have primary adrenal insufficiency and don't make cortisol. Cortisol is very important for dealing with stress and infection, but also maintains your normal blood glucose. And so they can become hypoglycemic. Their salt levels, the sodium levels can be low. And so those things are usually corrected by the addition of adrenal hormones, hydrocortisone supplementation. We know that the knockout mice are hypoglycemic and that also corrects with gene therapy. So we think that might be part of why they're dying so young, is that once they get weaned from their mother, they, it takes them a while to find the food chow in their new pens, and they may get hypoglycemic and just not be able to survive. So I don't know of any ketone abnormalities though.
Okay, thank you. And there's a comment by Alan Thomas. He says SGPL1, not to be confused with SGP7, because as I found a typing error in my medical notes. So I think the acronyms are a little complicated.
Yes, and there's an SGP1 that also has some kind of related phenotypes, and we had to be careful with that as well. SGPL1.
Yep. And I'm trained as a clinical pharmacologist. So part of my interest, I mean, when you do repurposing small molecules like the pyridoxal 5-phosphate, do you know if it is getting into the brain? Does it cross the blood-brain barrier? Do you see any improvement?
That's a really good question. I mean, I think it does because, people, neurologists treat infantile seizures with high dose pyridoxine. It's why we know that it's so safe in infants, because they use high dose pyridoxine. So if that's a neurologic manifestation that responds to high dose vitamin B6. So I'm pretty sure it gets in.
Okay. I mean, because sometimes dosing also matters. So, I mean, if you're not getting enough of the drug, after the first pass metabolism, it may not get in the brain enough. And
you're raising an important question that, I'm hoping to get I'm hoping to get an R01 funded. I'm like on the border right now of my funding, but it's a question that we could address with our mouse models. And I hadn't thought of that.
And if you can measure drug levels, you can actually look at how much drug is getting in and where it is getting in. Exactly. So we can always talk offsite, but there is a question from Zia for the panelists. Okay. Yep, that's for the general panelists, not for you. So thank you so much, Dr. Sarma. Again, too many windows to take care here. So look at a little sidetracked here. Thank you so much. So we'll move on to our next speaker, who is Professor Lara Bloom, who is the President and CEO of Ehlers-Danlos Society. And she's responsible for globally raising awareness of rare, chronic, and invisible diseases, specializing in Ehlers-Danlos syndrome and hypermobility spectrum disorders and related conditions. And she has coordinated medical collaborations, raising funds for research, focusing on the global progression of these two conditions. And she has offered to speak to increase awareness about these conditions to medical students and professionals. And thank you, Lara, for being here today with us to, again, educating us about these conditions. I need to point out that she was officially appointed as a professor of practice in patient engagement and global collaboration at Penn State College of Medicine in 2020. So Lara, please take it from here.
Thank you so much, and thank you for inviting me to speak today. It's a pleasure to join. I'm going to share my screen, so please bear with me. I think I've got the, can you see that, or can you see also my notes and everything else? Or can you just see the slide? Sorry, you're muted.
We see the notes.
We are seeing your notes.
Okay, I don't have any notes, but it's not as nice for you to view, so hang on a minute.
Mm-hmm.
I can guide you. You can just go to the full screen mode again. You can go to, and on the top, you can see swab display, the third option on the top right, left.
This
in
one?
The full screen.
No, in the
full screen.
Full screen mode.
Okay, if I do this then, and then what did you want me to click on?
On the top left, you'll see a swab display option, the third option.
Mm-hmm.
Perfect. There we go. Does that work?
Yes.
Perfect. Perfect.
Lovely. Thank you. Okay, so as I said, thank you so much for inviting me to talk today about the importance of patient engagement across research from bench to bedside, and really thinking about how we can ensure that patients are seen as equal stakeholders throughout the process. So trying to move my slide, let's try this way. My screen seems to have frozen. Let's have a look. I've got the spinning rainbow, which is exactly what you want. There we go, we're moving. I have no conflicts to declare, but you can see here I have a lot of different involvement in various activities. So I'm going to speak to you about patient engagement, and we see it across all of these different examples. Oh, we're now moving too fast. Hang on a second, I'll just go back. There we go. We know it's important in governance, in innovation, in evaluation, sharing activities, insights. It really needs to be thought of throughout everything that's done. And so often when it comes to patient engagement, patients are sent the final version so that people can say they've ticked a box to say that they've got the patient voice. And really what we are saying is that it needs to be right at the very beginning of any work that is done, that the patient narrative is brought forward. And also that the person, the patient, the caregiver, the parent is seen as an equal stakeholder. It's not as a token to sit a patient voice around the table. It really needs to be seen as equal. And I think we're getting better, we're getting there, but it's certainly not 100% of where it needs to be. As you heard in the introduction, quite a pioneering appointment was given by Penn State College of Medicine. I was extremely honored to be given the position of academic affiliate professor of practice in patient engagement and global collaboration. And finally, as the borders were open, I was able to do my first in-person lecture there just a week or so ago. And it was amazing to be able to speak to students and hear how needed this message really is. Phrases that, you know, people really thought they understood how important patient engagement was, and figured that if at some point in the duration of a study patients were involved, then you'd done right. But kind of still how wrong that was, and how comments patients equals data, and, you know, we'll check with the patients at the end of the study to check that they agree, where they think they're doing great, but actually there's a lot of improvement to be had. So I hope that this appointment brings forward a lot of progression in these areas. Okay, so ways that we're doing this at the Ehlers-Danlos Society. In 2018, we were awarded a grant by PCORI, which is Patient-Centered Outcome Research Initiatives. And that grant was to form the EDS and HSD Community Coalition. At that time, it was actually called the Comorbidity Coalition. And what that did was bring together stakeholders equally in a group that included largely patient organizations, individual patient voices, healthcare professionals, and across all the different types of EDS and HSD. And we worked on what we believed were the research priorities for the patient community, using the patient voices to bring forward that message. And they really did serve as the bridge between the community and the organization. And it was so successful that after the funding was finished, we actually have been funded by an individual donor going forward. And still today, this coalition is growing and thriving. And I'm also pleased to say that 2 of the 3 priorities that were identified have been funded, and we're still seeking funding for the last priority. And I think what this showed us was really bringing to the center what was important for the community, and not just what would be interesting for researchers to do. And we started with 95 different priorities, which was huge, and we managed to get it down to 3, which was a very big task. But I think hearing from the individuals themselves about what was important and how it should be done was really very, very important. Other areas that we've been using patients at the center and heart of everything is our AHEAD coalition. And that's Action for Infants, Children and Young People with HSD and EDS to ensure accurate diagnosis. So many in our community face a very long diagnostic odyssey, which is shared by many rare diseases, but especially when it comes to children. And not just are they missing diagnosis, but equally they are being accused of, parents are being accused of Munchausen's by proxy, fabricated illness, and over-medicalization. And it's been really causing a lot of problems in our community. And so we, again, used the Comorbidity and Community Coalition as a framework and did the same thing. We brought together other organizations working in this field, members of social care, judiciary, patient voices, parents, and healthcare professionals to work together on the best way that we can improve things. We also identified through that what research should be done to try and improve the outcomes for the pediatric group. And something that we often use and we have used for other studies is our global registry. Now, our global registry has just under 15,000 people in it from all over the world living with these conditions, which is pretty remarkable. And it's a self-reported patient-led survey which is now on REDCap, which really captures what individuals are going through and how they're impacted. And that's been hugely beneficial. And it's actually been through our global registry that we have been able to recruit for HEDGE. Now, HEDGE is our hypermobile Ehlers-Danlos genetic evaluation study. And it's really trailblazing in the sense that this wasn't led by an institution. This was led by us, a patient organization, because the parent of a patient approached us wanting to fund this study to try and find the genetic cause, the pathogenic variant behind hypermobile EDS. Out of the 14 types, it is the one type which we do not yet know the genetic cause for. And so we were donated $2 million to try and find the answer. And we led the whole thing. And because patients were at the heart of all the decision making, we were able to pivot through the pandemic and move from in-person enrollment to virtual. And so that it made us more inclusive, more diverse, more geographically spread. And that was led by patients telling us what was needed. So we arranged for blood draws to be taken at people's houses to ensure that the study still progressed and moved forward. And it's really been incredible. And we were targeted to reach 1,000 people by the end of this year. And I'm delighted to say that we would have been able to do that. And we wouldn't have been able to do that without the patients lending the voice at every single stage of this study. So I hope I've given you a few examples of ways that we're ensuring that patients are factored into every single stage of our work. I think it's essential that whenever you're considering a research study, you need to ensure that patients are around the table as equal stakeholders. Think about funding, think about time that they are giving. And it's not just, you're not doing them a favor by including them. You need to, if you're compensating other healthcare professionals, you need to also compensate the patient expert as well. If anyone has any questions at all, you can reach out to me. Here's my direct details, and we also have the details of the Ehlers-Danlos Society. You can find us pretty much on any platform, and I'd be happy to answer any questions you may have. Thank you.
Thank you, Lara. And it's such an important thing that we should be always engaging with patients. And I know in the US, the National Institute of Health is always promoting researchers to work with patients. And I just wanted to know, see if you had any insight on how we could do something similar in India, how can we engage patients better or promote patients to come forward and to be willing to talk to physicians or researchers?
I think it's a great question, and I don't think wherever you're based should be a limiting factor to doing that. And I think, as I said, if you are embarking on a study and you're putting together your plan, you need to ensure that at the beginning of it, the very, very start, you've included patients. And you look to organizations in your local area that have those connections with patient groups. Social media is an incredible tool. You know, you can use that to find patients that could be able to help you market through other umbrella organizations such as IndoUSrare, for example. I think there's always ways that you can connect patients, and you'd be surprised how many would be willing to help.
Yep. I don't see any other questions coming in, but I'm hoping that you can stay with us for the networking session. Probably there will be more questions where the audience can speak freely with you. Maybe that will be questions.
Unfortunately, I have another event in 15 minutes, so I'm here for 15 minutes, but then I have to leave, and I'm very sorry. But that's why I left my contact details. And if anyone wants to reach out to me anytime, I'd be happy to chat.
Thank you so much, Lara. I really appreciate you being here today. I know you have, again, a very busy schedule. Thank you.
No problem at all. Thank you so much.
Yep. So next we have Dr. Anita Hegde as our speaker. Dr. Hegde is a consultant in pediatric neurology at SRCC Narayana Healthcare Children's Hospital, Jaslok Hospital and Research Center, and Baijarbai Wadia Hospital for Children, and all of which based in Mumbai. And her fields of interest are in epilepsy, disability, and movement disorder. She's also the president of the Association of Clinical Neurologists, Child Neurologists in India. And today she's going to talk about her rare disease research, how she has been contributing to this field. Anita, thank you for being here. Please take it from here.
Thank you. Can I get to share my screen? It says it hasn't been. Okay. At the start, I feel very small and humbled by the talks that I've just heard before me. I think Lara, Vinod, Julie, what amazing work y'all are doing in your respective spaces. I have to claim at the start that I'm just a simple clinician with a very heavy practice in a city with a very big population. And while most of my work is clinical, yes, we like to work and do a little bit of research, but just to highlight the challenges that we face in research as well as advocacy with rare diseases. So what I'm going to cover is basically the challenges with rare disease research, the challenges with rare disease advocacy, and maybe some avenues that we could focus on. So just to give you an idea, been in research for the last 10 or 15 years with the hospital that I work in. Another disclaimer is that I don't work in a government, but I work largely in a private enterprise or a corporate hospital. So the funding that comes in for research is very different from what happens in government or central government, where they have access to grants and funds. So most of my studies have been international studies, which have been drug trials largely. A few of them have been epidemiology or registry studies. And these are just the 2 studies which I got maybe in the last 5 years, which were on rare diseases. Till then, rare diseases was never in the spectrum or never done. This is the studies that have come to me through the national system, where companies have come through, which from India itself. So you can see how small the percentage is relatively to the international spectrum. And the last ones were homegrown studies, where I've tried to do my own stuff with my institute through our own ethics and through our own scientific research board. So if I started in 2007, over the last 15 years, basically, I actually went through large numbers drug studies, was inspected by FDA and approved in 11. Hospital actually allocated a research room for me. And we may have just 3 or 4 or maybe maximum 5 physicians who actually do research in the entire institute. And rare diseases came onto my plate only as late as 2018. So I'm trying to highlight that in a busy clinical practice, and most of us, is that how small and how relatively unimportant research is in the Indian scenario. So very low research in the developing world. Clinical trials in India were actually stopped for a period of 2 to 3 years because the DCGI found there was some inappropriate stuff, and it was stalled and everything was held up and all our studies came to an end. Then it restarted again in 2017. And you can see we have a host of problems that we have. Ethics committee, every center does not have ethics committee. So many of the small centers, if you want to do something, you have to go to a public ethics committee. Training of staff for clinical trials, none of us are exposed to this as students and fellows, and you have to make the effort to learn this research over a period of time. So as I said, in undergrad, research is just not on our radar. In postgrad, your thesis is as much as you ever did in research. It's largely self-taught and learning through workshops and on the job. No formal training was given to us in research and academics. Hospitals and institutes are not equipped. I've actually had to learn that a research room has to have this thing where the drug is kept separate and you are kept separate. And we learn all this as we work on the job. And government hospitals do have to do much more research than private because their designation and their position and their promotions depend on it. But when something became compulsory, we've had our own issues and on originality and authenticity down the line. So what are the challenges with rare disease research? In particular, the central body research for rare disease is not officially from government point of view is really not a priority. We have so many acute medical conditions with large percentage of populations and number of people affected, that something where your percentage is small and your numbers are small is not going to catch your eye, other than when certain groups make the effort, do promotion, push it forward, and put it literally in front of the government's eyes to see that something happens. At the hospital level, we do not have the infrastructure and the workforce. And when you have a rare disease study, your numbers are relatively so small that the economics and the finance of it may not work out that well for everybody around. At the professional level, with populations that are big, research for rare disease is again not usually a priority. Epilepsy, the number of studies you get is huge. I've had 4 on ataxia, numerous other things, but rare disease relatively is difficult to find companies who want to invest so much on rare disease research. And surprisingly, when we did have the rare disease studies, the patients were the most skeptical and the most hesitant because of an innate fear that maybe we were just trying something on them in spite of all the efforts to allay the anxieties. The concept of research, as I said, not all understood the concept of research in rare diseases, and people are not so willing to come forward in it. So the challenges is lack of awareness. We have lack of infrastructure, prohibitive costs, which most Indian companies or institutes do not have the bandwidth to do. There are cultural influences. So it's like today I was speaking to someone and he said flatly no for some study I wanted to do, because he says the Almighty decides and it's not for me to decide. So we have a lot of cultural influences which guide our patients, and lack of government initiatives and fundings. So we are all aware of the Drug Controller of India and how everything has to go through it. And we now come to the challenges with the rare diseases and advocacy. Patients of rare diseases really do not even get to know or meet each other. There's very limited access to each other. If you do not have a support group, how do these people get to meet each other and connect, interact, and support each other? Very few rare disease support groups in the country. When you see the number of rare diseases, the groups are limited. People are sometimes actually, as I said, suspicious of joining these groups. The existing support groups are relatively basic. What do? They do connecting people, giving them comfort, explaining to them where they've done this or where they've done that, tying up with the support group, which may be abroad, but we don't have any research of our own in our rare disease groups. We are still at the basic stage of just identifying and putting the group together. We have limited focus on research. We have no government body for support for these groups. We have limited or no funding. It only comes from a few parents. We unfortunately do not have large philanthropic family donors who establish centers and research groups. If someone does philanthropy, usually that money is spent, as I said, on acute, urgent medical conditions with larger populations to benefit. There's limited awareness about disorders and limited awareness about treatment options, and the number of patients is a challenge for drug trials for rare diseases. So as I think it was Vinod who pointed out that a lot of time is lost, whereas in the US, it says it takes 7 years for a rare disease to be diagnosed. You can imagine how long it actually takes in India to diagnose a rare disease. Genetics has changed the way we look, but we do not have these umbrella organizations. Yes, we do have the ORDI now, which is the Organization of Rare Diseases of India, which is one to start with. But compared to the number of organizations you have in the West, we have a very small number. Most support groups have been started by parents of children suffering from them, and they put their own little finance into the system and start the program. So while we have Rett syndrome, and I know Mr. Sethi is on this, which started in 2010 and is doing a great job. Something as Angelman started literally 2 or 3 years ago, where we initiated and supported them, and it's picked up. So these are a few of those that we have. Some of them are neurological, but very few considering the number of rare neurological disorders we have. These are the rough support groups that exist at the moment with Rett's, Fragile X, Prader-Willi, Angelman's, storage disorders, SMAs, and DMDs. But there's a host, I can't even put the list down here, of those, even in neurology, forget other specialties, which do not have support for these parents. So how would you make it better? Could we do tie-ups with international support and advocacy groups who are running since a long time so that we don't try to reinvent the wheel? We benefit from the knowledge that they have got over the years. We can lean in and contribute with numbers. So when research is happening, just as Julie said, she would be interested. I am almost sure if we reached out to the nephrology fraternity, you would be able to find significant numbers of children with the gene abnormality you're looking for. If well-funded international groups can give some guidance and financial support to these advocacy groups, do tie-ups, do workshops, create support groups, help them to understand how to set up a support group, I think a lot can be achieved if someone who's already running something and has learned the ropes can support us over here. Conferences like this, I think it's amazing, fantastic. Medical symposiums on rare disease promotion, focus on academics, and research and increase awareness. I'll be honest, the sad part is those in the rare disease fraternity meet quite often and you all do a lot with each other. Now, I've entered this program today, and the first thing I realized is I didn't even know that Vinod was doing such amazing work sitting out in the country itself. So making clinicians who sometimes may not attend all these programs, which are very specific and aware of the fact, something like a directory. So if IndoUS Rare could help us set up some sort of directory of all those working in rare diseases or genetic aspects within the country, within the US, so that clinicians have a way to access them and refer to them. I think that would be a great tool as well. And a little bit on funding is always needed. Just to give you the figures of rare disorders that I may have. Single center, myself alone, single physician, over 1,200 whole exome sequencing done from my tertiary center with 760 cases, 60 plus cases positive, of which I have 200 plus epileptic encephalopathies. I just reviewed 175 movement disorders I want to present in a conference, 142 neuro, and the numbers are staggering when you look at it. This is one center from one physician in India. Can you imagine the numbers we could get now that genetics has come in if we could pool our data and analyze it together? So what do I do when I get a rare gene in front of me? I quickly go onto the net and I see in the NIH, is any research happening anywhere in the world on this topic? I'll see that gene therapy is starting somewhere. Something else is being tried in some other institute. I go on the net and I email that institute and see if I can put my patient in connection with them. Till date, all the patients I have tried to connect, not one has been able to avail of that. The only one is actually going for AADC gene therapy. We're trying to raise funds to send him across to Poland to see if that can happen, because most of this research is largely for the local population where this research is happening. So we know it's there. We do not have access to it. We do not have the ability to fund it ourselves. We do not have the ability to fund them to stay in those countries for 1 or 2 years while they go into that gene therapy. So while the world is moving forward and genetics has opened doors that are unexplainable, we still have a lot to try and make the world as equal as possible. So to give the family, so what I eventually do is I put them in contact with the support group and I try to keep up with the latest treatment options, which is there, and guide them to it. But there's a big gap or a gulf between the two. So what we have the numbers. We don't have access to extensive clinical and basic research, and I don't see it coming for a long time. We have more pending and important concerns, medical issues to deal with. We don't have the financial support. But I think platforms like we are on today are excellent starting points for us as clinicians to try and collaborate with research groups and see how our numbers, your financial support and research acumen can be put together. Thank you. And this is just my team that works with me, and without them, this would not be possible. Thank you.
Thank you so much.
Take any questions.
Thank you so much, Dr. Hegde, for excellent presentation and highlighting the various challenges that are unique to the Indian ecosystem, and also the opportunities and the good work that has been happening. We take note of your suggestion for a directory of resources and researchers in India and US that we could look up, and we will take that up as an action item from here, and many other action items as well. So time for a couple questions. If you can type your questions in the Q&A box. So there is a comment by Elizabeth McGee that, are you familiar with mytomorrows.com? It's a company, MyTomorrows. They may help with getting therapies.
No, I'm not familiar with them. Thank you. So that's what I'm saying. If we can put all these little contact points together so that clinicians can get access to this, thank you. I will look into it.
Great. Thank you very much. We are also upon time. We have, last but not the least, Nathan Peck, who will be presenting about Cure VCP. Let me introduce Nathan Peck. Nathan and Cure VCP Foundation has been a member of our Patient Alliance. Mr. Peck is the founder and CEO of Cure VCP Disease. It's a patient advocacy organization dedicated to driving a cure for Vallocin containing protein, VCP, associated multisystem proteinopathy, MSP, also known as IBM PFD, inclusion body myopathy, early onset Paget disease of bone and frontotemporal dementia. I have known Nathan personally, and incredible fighter, and his family is affected by this disease, and he's doing incredible work to help other people affected by this condition who go on to live in their adulthood with severe disabilities. So Nathan, over to you. Welcome.
All right, thank you, Harsha. Appreciate the nice introduction. IndoUSrare is a fantastic organization. We've enjoyed being a member really since the beginning, or early beginning. And thank you everybody for attending today. So what I wanted to do really quickly was just share with you 2 patient organization funded studies that we're leading for natural history. Okay, so real quick on VCP, won't spend a lot of time. I'm going to try to move as fast as possible. It's the Valicin containing protein, as Harsha said. We actually have an identity crisis. It's called different diseases: VCP disease, multisystem proteinopathy associated with VCP, and IBM PFD, which addresses the main phenotype symptoms. And so inclusion body myopathy, which is what I have, which is what my mother had, affects about 90% of the patients. Paget disease of bone, which my mother had, as well as the IBM, I don't have that. There's actually treatments for that if you catch it early enough. FTD, I think many people know about that, just, very cruel. Some patients develop ALS with this gene mutation. And so some develop Charcot-Marie tooth and some Parkinsonism. So very much a patient can get 1, 2, 3, or 4 of these symptoms, and it's not understood why that is. Just a real, it's adult onset. So I started developing symptoms in my late 30s. This is a, hold on, let me turn off this thing. Okay. This is a picture of my family. So it is, because it's autosomal dominant. This is my mother, my uncle with the disease, my other uncle with the disease, and an aunt that later developed the disease. And so this, it's ultra rare. We don't know how many people, we don't know what the prevalence is. We estimate less than 2,000 globally, but when you find families, you find the disease. And even between family members, as I described with my mother, you can have different symptoms, within the disease. Many patients get misdiagnosed. Okay, so Cure VCP disease, we were founded in 2018. We're entirely volunteer led. I'm actually on disability, but I lead the organization. We have a 5-person medical advisory board, a 9-member board of directors. And our goal at the start was just to identify patients and help the research community. But we've done a lot, in those coming up on 4 years now. I'm not going to go through in detail, but we were honored to be selected to the CZI Rares 1 Cohort 2 project in November. So kind of a validation of a lot of the work that we've done. So what I wanted to start with was, there are drugs that could be repurposed or have interest in VCP. One of them is Amylyx. It's an ALS drug. It had negative news about a year ago. Now, some things are changing. They reapplied to the FDA, but why is that? The patient community seemed to think it was working. We had Orphazyme, arimoclomol, which was tested in clinical trials in ALS, clinical trials in sporadic myositis. Again, negative news. And so from the patient perspective, my concern is why? Was it because of efficacy of the drug? Or more importantly, when I go to the clinic, these outcome measures that I do, I don't feel like, I know what I'm going through as a patient. I know what I'm feeling, but a lot of times I don't feel like when I fill out the IBM FRS, the functional rating scale, nothing changes. And so we had a need. So we have a patient registry with cords. Sophia talked about their use with cords. We have a little over 80 patients. It's a global registry. We have none from India, though we have spoken to 2 researchers via IndoUSrare that say they have VCP patients, but genetic testing is a big issue. We're doing a retrospective natural history study with Allstripes. We're doing a prospective natural history study. We have funded this using traditional measures, with Nationwide Children's Hospital. And so this is more your traditional prospective study, and I'll talk about that in just a second. But we're working with a company called Casimir that is started by a couple of Duchenne muscular dystrophy parents, which is more focused on video measures and more sensitivity in those measures. And so what I'm going to highlight and talk about very quickly is we're funding both of these studies with these companies or organizations and how we're approaching that. All right, so this is the nationwide study. And so this is how it's a very traditional natural history study with outcome measures. We're using the North Star. For those of you familiar with the neuromuscular, we've focused on the neuromuscular piece. We're not focused on the FTD because we don't want to cloud and pollute the data set. Most of our patients have neuromuscular symptoms, so we're focused on that. But we have the North Star, the timed up and go, very traditional measures. A lot of these measures, for the upper extremity, we're using the pull, hand grip strength, 9-hole peg test. Again, very traditional measures that have 3-point scales. So it's very hard that, one thing, ALS is a very fast, rapidly moving disease. My disease looks a lot like ALS, but it's over a much longer time horizon. My mother dealt with it for 20 years. I will deal with it for 20 years, or, who knows? But, it's very slow progressing. And so it's hard to have sensitivity and to notice if things are changing in those measures. We're doing some spirometry. We're also doing patient reported outcomes via our registry, as well as other surveys. There was a question earlier about how we're reaching it. So this study is actually done in clinic and it's done remotely as well. And I'll show that in a second, but we, as Cure VCP Disease, put together a patient kit that we have mailed out to all the participants. We have about over 25 participants in this study. Again, ultra rare, so it's hard to get people to sign up. And there is a stigma with some patients. Why you want to do this? Why you want my data? Which we're trying to address that by getting them to participate. So this is, again, very traditional study. It's over 12 months. We're a little concerned, will we see change in the study? Over that 12-month period, will we be able to notice change? So we're working with a company called Casimir, and many of you may be familiar with them. And we're doing 14 different independence tasks that we will put on video. So the patient can do this from the comfort of their home. And then PTs will actually score this along a 15 or 19-point scale. That's actually their intellectual property, how they do this, how they do the scorecards. But I wanted to show you briefly, so versus just kind of lifting a cup or doing a dynamometer test, here is some videos.
When I say go, pick up the sock. Ready, go.
Okay. So that's actually probably the scariest one I did. I was worried I was going to fall because my quads are dead. They're just kind of gone. But that video will be scored at time point 0. It will then be at another time point scored. I'll do it again. And then at another time point. And we will see across those sensitive measures if we're able to tell a difference. So this is me getting off the couch.
Stand up until you're fully upright. Ready, go.
So how I have to compensate myself, that's what's being scored and measured. And then I'll just show one last one.
When I say go, stand up until you try not to rest against the back of the chair. Start with your arms at your sides. Make a circle with your arms until your arms are straight above your head, next to your ears, if you can. Try not to bend your elbows. Ready, go.
All right. Okay, and then we have another one. This is a side view of that same one so that the PTs can see. I actually have a lot of scapular winging, so that's very apparent in this video. And then even rolling over in bed. And so while we find these valuable is these are independence measures. These are things that I'm losing my independence, but these are very important to be able to measure, because these are everyday activities. So, oops. Okay, so is there a sensitivity difference? So we're not competing. These organizations are actually working together on this, and we've funded both of them. So there is no competition because we need to understand in VCP disease, can we tell a difference in 12 months? And so nationwide, at the start of the study, it was in clinic and remote. We were actually testing the difference. Was there a variability or a difference between being in clinic and remote? And at time point one, we're already at 6 months in that study. It doesn't appear there is, which is great news. So everybody does it remotely. And then we funded everybody, the 25 to 30 patients to go back to the clinic. They will go back to the clinic in 12 months. So at the same time, we have Casimir. So Casimir has started their study at 6 months. We did 2 measurements at the start and then remote once and remote in another period. So we'll have 2 periods that kind of overlap. So that's very exciting to us because we can kind of see, did they notice a difference versus what the traditional study noticed?
Last minute alert, Nathan.
Do what?
Last minute alert.
Okay. So, again, it's not a competition. We won't have results published until January 2023. Patients, they own the data. So data ownership is really important, though the institutions have a right to the data. But we feel like this is useful for many different diseases, including limb-girdle muscular dystrophy, myositis. So we don't want to do this just for ourselves. We want this to be a great scientific endeavor for a lot of different diseases. And so that's what I have. I'd be happy to answer questions at the end, but we're very excited about what we've been able to accomplish and fund and do.
Fantastic. Thank you so much, Nathan. We have time for question and answers, as well as we can move into the networking session as well. So Sadiq, if you can enable everyone to join the session. Any questions for Nathan, you can ask directly or you can type in your question. To conclude today's session, we had some amazing speakers this morning. Really enjoyed the insightful, eye-opening remarks made by every speaker today, both from Indian side as well as the US side. Great exchange, and this is what we really love to see happen more often. And we will endeavor to take notes and follow up on all the suggestions given by all the speakers today. So the floor is now open for networking. We will regroup again tomorrow for the remaining 2 days of the program. Tomorrow, we are going to have all the patient alliance members speaking. There is going to be a series of patient foundations making presentations about how they have gone about their own disease and community, how to help them. And the final day on Thursday will be about Industry Day. We will have a lot of speakers from the biotech pharma industry and a fireside chat with a legend, Frank Sasinowski. So stay tuned and let's start with the networking for the rest of the session today.
Attendees can now unmute themselves and talk.
Yeah. So any questions for Nathan? I personally, I've been a huge fan of Nathan and appreciate his friendship and collaboration as a rare advocate myself, having been a rare dad. I admire the work and tenacity that Nathan demonstrates. It's an inspiration for many of us.
Appreciate it, Harsha. You've been a wonderful supporter of us. Thanks.
Welcome. So the floor is now open for open discussions, Q&A. Feel free to chime in. Dr. Sharma, do you have a comment?
Hello.
Yes, please go ahead.
Yeah, this is Manoj from India. I would like to know, like how this, Indian, the people in India, the rare disease patients who doesn't know, there's a lack of, lot of difference in diagnosing the disease. How the genomic sequencing thing will reach to the common man? Because until unless he doesn't undergo through a genomic sequence or a whole genome sequence, he would not be able to know whether it's a rare disease and how rare it is.
Great question, Manoj. I think Dr. Vinod Skaria and Dr. Tangaraj would have been better people to respond to that question. They unfortunately have had to drop off to their next thing. But my perspective is that there is not necessarily a need for whole genome sequencing right away for all the cases. Even a targeted hotspot panel, like Dr. Skaria mentioned, where they have optimized that panel for the Indian population and made it available more widely at a low affordable cost. That is already useful for diagnosing many of the known genetic diseases. Now, for many of the yet diagnosed or yet to be identified new diseases, which do happen, about 200 new genetic diseases are being discovered every year as a result of the advances in genetics and genomics. And for those yet unknown, whole genome or whole exome sequencing can be more useful. But for all the known diseases, the hotspot panel is both affordable and more accessible. We receive requests from many patients in India who come to us with the report already in hand and seeking assistance.
Yes, Harsha. Right now, I'm like, we are working. I don't know if I am a patient or a caregiver, because one of my cousin is suffering with a disease known to be HAE, hereditary angioedema. But we want to clinically collaborate it, such that as because the symptoms are same, but whether this is the same disease or not, because due to the cultural differences, people are not ready to accept it that they are suffering with that disease. Until unless we have given our samples twice at CDFD, and even now we sent our samples to PGIMR Chandigarh. It's taking a whole lot of time, and I even inquired with a few of the private testing facilities. They are charging about somewhere around 3,000 30,000 Indian rupees. But given the condition of the patient, economical conditions, is not in a position to bear.
Yeah, I hear you, Manoj. I will say that it's still a lot more affordable with 30,000 rupees cost in India compared to what people pay in US and other countries. So it's still expensive. It's not come down as much. Hopefully the cost will go down as well and make it more affordable. I don't know, Dr. Hegde, did you have any thoughts for Manoj?
Actually, if you see the cost of genetics today in India, it may be cheaper than anywhere else in the world.
Yeah.
Because of the numbers, what it started off 5 years ago, today it's like, compared to the West, it's almost throwaway price. And we are getting bigger and bigger panels of WES with microarray, with everything connected together. I think with time, the cost will even come down further. And I think I doubt insurance will ever cover it, because they don't cover anything genetically or congenitally driven. So I doubt that will ever happen.
Yes, that's an issue.
Yeah, I can't see that happening, because any of my patients who have something genetic or with the word congenital or from the word from birth written, no insurance company is willing to cover them. But I think the cost will come down even further with time. And yes, I suppose everybody is not going to get access to it, but that way everybody doesn't get access to a doctor who knows all about the genetics either. So it's multiple things have to fall into place for everyone to get the benefit.
Yeah. And Manoj, we can connect offline and see how we can assist you with information and resources.
Sure. Thank you.
Anyone else likes to make a comment, introduce yourself, and maybe ask a question or make a comment?
Yeah. Hey, Harsha, it's Brian Toton.
Hi, Brian.
Hey, it's really good to see you again. And I know that we had this conversation, a couple of weeks ago. One thing that I think is going to really be, it's going to really help the rare disease detection movement is really the incorporation of better microbial imbalance monitoring. So if we can do more monitoring that way, a lot of times, I mean, like 95% of all illnesses stem from inflammation. So if you know it isn't inflammation, you can pretty much determine that it is probably something more difficult to detect. And then I think when that progression of, answers comes to be fulfilled, you don't have these patients that spend, 5-10 years trying to figure out, kind of like the underlying cause of their, their illness. It's really just, being accompanied with more routine monitoring that I think is going to really help, with the balance. So, as you guys progress, I would definitely say, incorporate yourselves with more, basic monitoring platforms that do kind of like a shotgun of, what a systemic illness would be traditionally. And, when it doesn't fall in that category, then it's almost like, well, we need to do more whole genome sequencing, identifications, and then work with like bioinformatics services to kind of identify specific pain points within the genome itself that would identify, hey, this is a rare disease. I think we need to go in this progression.
Thank you so much for those comments, Brian. I'm curious if anyone has any opinion or perspective on this. Brian comes from microbiome and inflammation background, and his perspective is that a lot of the diseases stem from imbalance in the microbiome in different organ systems, as well as inflammation. I'm not sure how much evidence there is to say that, but wonder if what other people may be seeing in that space.
Harsha?
Yes, Zia.
Zia.
Yes, Zia, please go ahead.
My compliments on the great start and absolutely interesting conversation for 2 days going on. And see, my question, which I raised, is a general question. I put, it was actually Vinod, I think Dr. Vinod replied to that. It was just a general thing. See, actually, with my journey, with past 12 years or something, what started off with ataxia, what Manoj was saying about the cost factor and the genetic testing and where to get it done and all that. So about 12 years back, it was a very tough time for us, going through all the hospitals, doing the same SCA tests everywhere, getting it repeated at very high costs. And one of the questions which I kept asking at all the hospitals was that I'm coming over here, giving you the blood and getting the tests done for my son. Now, if there is some development happening, do you inform me? Do you tell me there is some research happening or some medicine being done? None of the hospitals had an answer to tell me that, yes, we will contact you and inform you that there is something happening. It meant that, once the test is done and it is positive or negative, the patient or the person is sent back home. That's it. Yeah. And the onus comes on the person to again go back to the hospital and keep checking with the same doctor to find out whether there's something happening. Yeah. So this kept on taking us from one place to another, one place to another. This went on going on like that. And when the registries also started, there wasn't much of information being collected, or I don't know who's having the data on this particular disorder like ataxia. Now, if I want to find out, in general, if I want to find out if there is some research happening or there's some medicine being developed or something, how do they reach out and inform the set of persons who have already reached out to them? So there's no data with them. It is, I have to keep on checking up with all the 10 or 12 doctors if there is some development happening. So what is the approach for that in this particular scenario?
So great question, Zia. I'll request Dr. Hegde to chime in again on this one.
It's not an easy situation. I have the same problem. So today, again, a patient comes with DMD and says, doctor, isn't there anything new? Last time you said you'll tell me if there's anything. But if there isn't anything, as a clinician, what can I send out to you? I know you, as all patients, are waiting and hoping to hear from your physician that something has come out. But very little comes out which is game changing at this given. I mean, it's there, and it's hopefully we're going to be more soon. But from our end of the side as well, I keep the notes, I have data, I have all the names and contacts of all the people with different conditions. Like when SMA came out, we connected with all the SMA patients and told them, listen, these are the 3 options. If you're interested in any of this, you want me to write for you or support you in whatever, please come over and we'll do it. But treatments like SMA treatments don't come out that easily and that often. So I can understand your concern and I can understand you wanting to be updated regularly. But sometimes I think you will know more about your condition than even I will know.
Yeah, if I can, if I can jump in real quick, Dr. Hegde, I'll help you here. That is the role of a patient advocacy organization. And I understand your frustration, but nothing, you know, we didn't know what was going on in my disease until we got started. And it required us plugging in and helping the Dr. Hegdes of the world, you know, that we actually organized a scientific conference back in September to bring, because a lot of these people don't talk to each other. And so that's, I just, you know, it's frustrating being patient. Trust me, I get plenty frustrated and angry, you know, many a day, but being plugged in and communicating and helping, you know, it's not about what they can give to you. It's about what you can give to them. What support can you provide? Because they're overwhelmed and busy. That, you know, that may not be the answer you want, but that's why patient advocacy is so important, because things have kind of flipped now. We're driving the agenda, you know, at this point, and the researchers and clinicians and doctors now understand that they need to involve us because we can help them. We can help them with a lot of this stuff. So I hear you. It's very frustrating watching and waiting, but that's why it's so important that you get involved and drive the agenda.
Yeah, great point. Nisha, did you have a comment?
Doctor, that's a nice point which you told me, that, you know, the doctors may not have the full information to give it to a patient. The scenario here in India, as you know, that there aren't much, you know, the maximum number of people who come are coming from a little economically weaker division or sections, and not many would be determined to keep following it up, because here the rate of people giving up is more in India, I would say. That's what I have seen. So people do pursue for some time and then give up after some time, and the reach out to the patient is it's going to be very, very difficult. That's what I've been seeing the scenario over here.
Nisha, did you have a comment?
Yeah, I think it was along the same lines, in that many a times there might not, as Dr. Hegde said, there might not be developments that are significant enough to be you know, communicated to the patients, but often the doctors themselves are not aware, even if there might not be. Dr. Hegde, you are, I mean, you take the effort to keep track, but many doctors do not have the bandwidth to do so. And this might be because of the inherent system problem here. How do we address this? That doctors themselves are keeping abreast of the latest in terms of research. Again, connecting with basic science as well as translational research. And also, when you ask patients to speak with the doctors, how to make doctors more open to listening to the patient's voice. This is again a problem that we have in India, as you may be aware.
So, you know, practicality, I would be honest, is there's so much happening in science at the moment, especially with the advent of genetics. So because of the understanding of genetics, so much is happening in different spaces and different fields simultaneously. It's very difficult for a doctor with a busy practice who's covering many bases. We are not just into one space, we're covering many bases. It's very difficult to be totally knowledgeable about the latest in every subject under your sphere to the utmost that is available. So one, I open up and look, but I'll be very honest, I actually do what you all mentioned. I tell the parent or the patient, the parent of the child, that, you know, over a period of time, you are going to be teaching me more about the subject that I am going to know myself. And if you want me to help you connect with a system or a center or a whatever, put me on the CC of your email and I will connect with your doctor to give them the information. But it's humanly impossible when you're to know everything about everything that's happening with the latest going on.
Absolutely.
So I sympathize with the patient totally. And I know, as you just said, that as Nisha just said, that the changes may not be enough to explain to a patient. And sometimes if I give the small changes and the small research to the patient, he doesn't want to know more research is happening and something new is being worked on. He wants to know what is in for me. How is it going to change me? So it has to be put across where you give him hope and you tell him that stuff is happening, but at the same time, a realistic picture is also needed.
Yeah, I will add to that from my perspective. Healthcare has always been episodic, right? Whenever a patient has a problem, the patient goes to the doctor. That's when the doctor gets involved in assessing the patient at that moment and providing a treatment option. Now, the continuous monitoring, continuous care is just starting, right? To my knowledge, it's not where you have a wearable sensor or remote monitoring of patients. That's just getting started and to be adopted. When that happens, there is opportunity to do more continuous update based on data analysis and dashboards that the doctors can see for each patient. That's where, when Dr. Hegde said humanly not possible, we need machine or automation and the AI, ML, as well as the technology-based approach for continuous monitoring. What we are planning to do with the patient concierge is a technology-enabled platform where patients who come with rare diseases making a request, we can set alerts and triggers where we can look for clinical trial options. For example, if there is a new clinical trial related to ataxia that is announced somewhere in the world, that can trigger a alert. Those types of things, that's what is being planned. So, as individual doctors may not be aware of all the clinical trials, the patient concierge that we put together can help that. And then the patient can then go to the doctor and say, I need your help in assessing whether I might be eligible for this clinical trial. That's how we see this progressing, Zia. Hopefully all of these combined comments can help.
Yeah, thanks, Arja, because you've been seeing these developments happening in the recent times, and it's a great work which you people are doing. So what we started off at that time was it was a kind of a dark periods where we were going groping around looking for some kind of an information, and like how Alan also has typed out, now it is the patient himself who has to do the study.
Yeah.
So we, in fact, we were doing all the researches, and we started finding out which doctor to attend and which hospital to go and all that. We ourselves decided and started meeting. A lot of development is happening. Let's keep our fingers crossed. One thing Dr. Hegde was also pointing out is, the most of the patients or most of the persons who come over there, they come looking for a cure, and that is the biggest trouble. That's where the trouble starts. The person is not interested in listening what is the future developments happening. They want a cure today. That is the problem.
That's right. That's one important lesson for me, having participated in patient advocacy in the US with various patient groups, is that patients have to take charge of their own health. And because every patient with rare disease will see number of doctors. I don't know what that average number is. They are referred from one doctor to another specialist, and they will end up seeing multiple specialists before they even get a diagnosis. And even after getting the diagnosis, you are still managing multiple consultations with different doctors to manage different aspects of the disease, as these diseases can affect multiple organ systems. And hence patient becomes the driver of his or her own healthcare as well as research. And enrolling in a clinical trial might be the best thing that can happen to a patient with rare diseases, in my personal opinion, because that almost always advances the standard of care. A clinical trial is only allowed if it has the benefit of providing greater benefit compared to standard of care. And hence, in rare diseases, that's probably the best thing that can happen to the patient. Any other final comments? We have run over time and we should conclude soon, but any closing burning comment or question? I know it's over 11 p.m. in India.
If I may, I have one quick question.
Sure.
I'm with Care for Rare America. We started in Germany in 2009, and we're just started a sister organization here in the US this year. So I'm a director of strategic relationship management and global collaborations. And our founder, who's in Germany, has spoken a lot about an interconnected database. And I'm just wondering, for other kind of research driven, also patient driven, we're more research driven than patient driven at this point in our growth. But when he wants to start a directory of this similar nature, how do you envision different organizations such as ours coming together if they do? Because I see this as the kind of the crux, different organizations coming together to contribute to these directories. How do you kind of envision this? And I know that's a hard question, so I may have to follow up after, but I was interested to hear your final thoughts on that before we.
Great question, Hannah. Technically, it's kind of like a Wikipedia page, right? Or a portal such as Wikipedia, where there are external contributors who can add new resources, new individuals, experts in different disease areas, and the directory keeps growing organically with users actually contributing data itself to this directory. And then ability to have rating scale, right, where different users can rate based on their experience which resource was actually useful for ataxia. Something that may not be very useful for ataxia, maybe it was useful for another disease, right, and so on. So I think it has to be curated and organically grow over a period of time, get sorted out, and with rating, and that's I think what needs to happen. Now, funding and how we find the resources to make that happen, that's something we need to discuss, right? Any other thoughts on that?
No, I agree. This was great. I think I'll follow up with you after the conference, but my brain is constantly going towards this direction and how we can all work together to achieve this, because I think it will take consortium and collaboration. Thank you so much.
You as well. Thanks for making that comment. A very important one to follow. Okay, I know it's very late in the night in India, and appreciate everyone staying late and all the discussions and contribution today. Very excited for various collaborations and action items. So we will be in touch with each one of you on our next steps. Thanks again. See you tomorrow morning, same time for the Patient Alliance Day.